Hi everyone, welcome to my poster on machine learning based identification of discriminatory microbial features for the classification of a diarrheal gut microbiota. We all know that diarrhea is a common disease with simple and effective treatment strategies, but unfortunately it is the second most common cause for the deaths of children aged less than 5 years. And nearly 75% of these mortality is observed in just 15 countries from Africa and Southeast Asia. Apart from the fatal consequences, diarrheal reoccurrence is also associated with the malnutrition incidence in children through gut microbial dysbiosis. Gut microbiota is the dense among all the human microbiota and involved in critical functions such as host immune system development, production of secondary metabolites, and digestion. Despite their significant role, it also has a high interpersonal variation due to factors such as diet, lifestyle, antibiotic uptake, mode of birth, and geographical locations. When a pathogen invades the microbial community, it could disrupt the community structure and results in reduction in the microbiota diversity. This state of reduced microbial diversity is called as dysbiotic state. Previous studies demonstrated that any disturbance during the gut microbiota development in early life could lead to microbiota immaturation and associated with long-term disorders. So it is important to understand the variation in microbiota composition upon diarrheal infection to treat and promote better gut health. For this study, I took data of healthy and diarrheal infected children from Mali, Gambia, Kenya, and Bangladesh from public databases. The 16S rRNA sequences were processed through CHIME to obtain the relative abundance table, which gives information about the species composition in the community. Three different approaches were employed to understand the gut microbiota variation. Firstly, microbial species that are differentially abundant between healthy and diarrheal cases were identified using DEC2. PyCrest was used to analyze the differentially abundant functional pathways between different host conditions. Finally, discriminatory features, that is, microbial species which could classify the healthy samples from diarrhea-infected samples were identified using machine learning methods such as iterative random forest and lasso classification methods. Features with a higher importance scores from RF and higher coefficients from lasso were selected, and the commonly identified microbial species were used for the classification. Moving on to the results, initial Permanova analysis showed that age and country is having a higher impact on gut microbiota composition than the host conditions like diarrhea or any other disease conditions. So all the analysis were done on the age stratified country specific dataset. Differential and abundance analysis exhibited that certain genus were associated with the host condition. For instance, Brevotella and bacterioids were abundant in healthy controls, and Shigella and Escherichia were associated with the diarrheal cases. It also showed the effect of diet on gut microbiota with a higher Brevotella abundance in African countries. Then the functional pathways involved in carbon degradation and fermentation were found to be enriched in controls, and it is in line with the results obtained from the differential abundance analysis. Whereas enrichment of amino acid biosynthesis were observed in diarrheal conditions as like in the other dispersed state. Finally, RF and Lasso were identified a set of 36 microbial species as a discriminatory features for the classification of diarrheal samples from the healthy sample. This analysis showed country-specific and age group-specific variation along with the condition-specific variation. For example, age group specific association, that is, the association with the host condition varies with age groups like Bacteroides fragilis were associated with the cases in the first year of life, whereas it is associated with controls after the second year. To conclude, the validation of these discriminatory features were done for the external datasets, and it could be a good starting point to analyze the metabolic interaction variations which could provide insights about the gut microbiome resilience and the development in early life. Thank you.